Recombinant DNA technology can be defined as a method in which DNA molecule made in vitro with segments from different sources or by combining genes from different organisms. There are few importance of recombinant DNA technology. The first one is gene structure, splicing and transcriptional regulation. The second one is protein function, domain structure, post-translation modification, phosphorylation sites. The third one is biotechnology, insulin growth hormone, gene shuffling, and the last one is gene therapy. Example of recombinant DNA molecules that are important to human are pharmaceutical like human insulin and antibiotic. The human insulin gene was recombined with bacterial DNA so that we can easily and safely generate large amount of insulin.
There are 5 tools used in recombinant DNA technology, which is first, target DNA that containing gene of interest. Second, restriction enzyme, which produce 2 types of cuts, which is sticky end and blunt end. And the examples of restriction enzyme is EcoR1, SMA1, and BAMH1. Third, DNA cloning vector. The examples of it is plasmid, bacteriophage, cosmid, and YAC. Fourth is host cell, which has unique characteristic. And last one is modifying enzymes, which has examples DNA ligase and type polymerase. So this is 5 tools in gene cloning. The first tool is target DNA. Target DNA is a sequence of DNA containing gene of interest. What is gene of interest? Gene of interest is one such gene which confer the desired phenotype. For example, antibiotic resistance genes of bacteria. The example of target DNA is gene encode for protein, which can dissolve blood clots, used in heart therapy.
Restriction enzyme is an enzyme produced chiefly by certain bacteria that has the property of cleaving DNA molecules at or near a specific sequence of bases. In other words, to easily remember, restriction enzyme cuts DNA molecules at a restriction site and produce short restriction fragments.
The third tool used in recombinant DNA technology is DNA cloning vector. It can carry foreign DNA into a cell and replicate in that cells. Its characteristics is able to accept foreign DNA in MCS, which is multiple cloning sites. Second, can replicate freely in the host cell. And the third is possesses selectable genetic marker. Example for DNA cloning vector are plasmid, bacteriophage, cosmid, and YAC.
Host cell, basically a cell which receives recombinant DNA and allow the replication of recombinant DNA. In other words, an animal or plant on or in which parasite or commensal organism live or living cell in which virus multiplies.
There are three characteristics of host cell that we should know. First one is able to receive. Able to receive recombinant DNA through transformation process. Second is able to maintain. Able to maintain the structure of recombinant DNA from one generation to another. The third one is able to amplify. Able to amplify the gene product from the recombinant DNA. Bacteria is the most commonly used as host cell. The reason why is DNA can be easily isolated from or introduced into a bacterium. Bacterial cultures grow quickly, rapidly replicating any foreign genes they carry. Example, E. coli.
Modify enzyme. It divides into two parts, which is DNA ligase and Taq polymerase. The DNA ligase joins the foreign DNA fragments containing gene of interest and bacterial plasmid by catalyzing the formation of phosphodiester bond to form recombinant DNA, Taq polymerase. It is used in polymerase chain reaction, PCR. It replaces the DNA polymerase because of the temperature conditions of PCR. Taq polymerase is to add nucleotides to the 3' end of primer in order to produce new DNA strand.
Step in gene cloning by using plasmid as a vector. There is 5 steps in gene cloning. First is isolation of gene. Second is cleavage or cut. Third, insertion. Fourth, transformation. And 5 is screening. First, isolation of gene. In this process, the plasmid from bacteria cell is isolated and the gene of interest is isolated from DNA donor cell. Next is cleave or cut. By using compatible restriction enzyme, DNA donor and plasmid bacterial is cleaved. It made a single cut within leg Z gene. Then it will produce complementary sticky end. Then insertion. In this process, the DNA fragment is inserted into the plasmid. They will join together by DNA ligase and form recombinant DNA. As the bacteria was produced, the recombinant DNA is also replicated. Fourth is transformation. The recombinant DNA then inserted into the bacteria through the transformation process. The bacteria will plate on agar containing antibiotic ampicillin and sugar, X-gal. It was incubate anticlonus grows. Finally, screening. Blue-white screening is done to identify bacteria clone containing recombinant DNA.